We'll now move on to the second presentation in the workshop, which is going to be given by Dr. Yojiro Yamanaka of the Goodman Cancer Center. It's going to be on the CRISPR knockout models, which Bill has been talking about relative to recent developments. So Yojiro actually did his PhD at Osaka University in Japan. And he was working on the interleukin-6 signal transduction, particularly relative to leukemic cell lines. After finishing his PhD, he actually stayed on as a postdoctoral fellow for 1 year at Osaka, became an assistant professor at Osaka, probably at the same time as he was trying to organize his move for a second postdoctoral training period with Dr. Janet Rosant at the Samuel Lunenfeld Research Institute at Mount Sinai Hospital in Toronto, where he made some seminal discoveries there. He stayed on after his postdoctoral for a couple of years as a research associate, and then essentially joined the Goodman Cancer Research Center as an assistant professor in 2009. He's now an associate professor in the same center. And Yojiro comes with a very in-depth training on stem cells and lineage specification in pre-implantation embryos, and was very much involved in the development of the CRISPR-Cas9 engineered cell lines in the Goodman Cancer Center, his own lab, of course, but also in the Goodman Cancer Center. He is director, co-director with Mitra Cohen of the transgenic facility, which now actually is called the MICAM. Mitra will talk to you about all the possibilities that exist in this mouse facility. And we're very happy that Yojiro accepted to present today. And Yojiro, the floor is yours.
Okay, thank you, and thanks for having me. Today, I'm going to talk about the CRISPR mouse model of cancer. And everybody can hear me, all right? Okay. So CRISPR, I don't think we need to explain the detail, but in this year, 2020, the Nobel Prize in Chemistry goes to those two scientists, Emmanuel Charpentier and then Dr. Jennifer Doudna. They developed novel genetic scissors, a tool for rewriting the code of life. This is a great discovery, and then it's really revolutionized the way we dealing with genome. And then CRISPR, and then I believe that all of you knows, is originally identified in the immune system in the bacteria. In bacteria, they used 3 components: Cas9 protein, this is the endonuclease, and then CRISPR RNA, and then transactivating RNA. In the bacteria, combining these 3 components, these 2 RNA hybridize and then forming complex and then recruit the Cas9 to the target site. And then this endonuclease can digest those double-strand DNA and then introduce double-strand DNA break. And then what those 2 scientists really discovered is really proved that that's only 3 components is sufficient to make this DNA double-strand break, and it would potentially work in a eukaryotic cell. To make the CRISPR system work in a eukaryotic cell, you need to place a nuclear localization signal to this Cas9 protein, otherwise they don't go into the nucleus. And then what is great in the kind of study is it doesn't need to have 2 RNAs, just to fuse this RNA, and then still sufficient to recruit this Cas9 protein to the targeting site. This is now called the single guide RNA, I'm sorry. And then what brings to the field is now we can modify targeted somatic, the kind of targeted somatic genome manipulation is now become possible. Is this the first technology? No. indeed, CRISPR is discovered around 2012 or 13, and around from the 2008 and 9, engineered endonuclease, targeted endonuclease start to develop and has been used in the field. Zinc finger endonuclease talents are the kind of, it's originally come in the field, and it's really started changing the way we modify the genome. But what was really difficult for us to kind of really use these two endonucleases is because they use the protein DNA interaction to detect the specific sequence. So it is difficult or not easy to design a new enzyme, a new endonuclease targeting to the novel site. Once you design, once you validate, the specificity or the function is quite similar to the CRISPR-Cas, but it was quite difficult to design the new one. Instead, in the CRISPR-Cas, what is really revolutionized because of that they used RNA-DNA hybridization or interaction to recognize specific DNA sequence. So it is easy to change this single guide RNA sequence that is sufficient to change the targeting site. So that makes kind of easy to generate a new CRISPR targeting a novel site-specific site. What all these three enzymes really do in a cell is introducing a DNA double-strand break at the specific unique sequence. If the no donor sequence is available, like a G1 phase, non-homologous end joining happen, and then occasionally it can induce the mutation called a small insertion and the deletion. If the donor sequence is available, they use a homology-directed repair and then completely fix those sequence. Or if you want to introduce a specific mutation and provide the donor DNA, you can introduce a specific modification to the genome. So before we jump into the kind of model, I'd like you to think about what are we modeling in the mice? Using those, what the cancer model is really modeling. And then I'd like to ask you, what are the sufficient conditions to generate malignant cancer cells from normal cells? I'd like to think about that. Do we know the sufficient condition to make the normal cell to the malignant cancer cell? Probably you may say, what's driving a normal cell to become a cancer cell are mutation. Is this sufficient? Probably not. It is not insufficient. And then some of you may know that the reprogramming, now we can make the normal cell or those fully differentiated cell back to the pluripotent state. It's called the induced pluripotent stem cell. You can induce those reprogramming, but the frequency to induce the reprogramming is quite low. It's 1 in 10,000, 100,000, maybe even less. Similar to those reprogramming, mutation alone is not sufficient to make the normal cell to fully malignant. This is a kind of evolutionary trajectory from the normal cell to become cancer. This is the kind of hypothetical situation, but gradually we start to recognize this is the case. Invasive neoplasm cancer from this kind of last stage, and then we believe that is starting from the clonal population. But when you look back, years or the decades before the cancer image, or the soon after the conception, you start to acquiring the mutation. Some mutations gradually expand as a clone, but not necessarily transform. And then mutation, what the mutation does is up to here. And then calcidogenesis or the transformation is not sufficient by the mutation. What needs to happen is a catastrophic change, like copy number alteration or the aneuploidy. That's happened around here. And then what we are modeling in mice is we are creating the situation like here to adding the mutation, and then but not we don't have a still tool to initiate the catastrophic changes, but kind of susceptible population for this catastrophic change. Some cases, introducing a mutation means creating these blue population. Some other case is this orange population that reflecting those latency and then the penetrance of those carcinogenesis in the mouse model. Then, what CRISPR bring to those cancer modeling? CRISPR can be used in various types of the mouse cancer model. One type is a transplantation model. People use some organoid normal cell or the cancer cell line or the PDX, and then modify their genome using CRISPR and then transplant to form those cancer model. CRISPR can be used to modify specific gene in those normal or the already cancerous cell. And also, CRISPR can be used to modify the germ lineage to make a traditional germline GMM. That's what Bill talked about. We can directly modify in the zygote. We can still use embryonic stem cell, but those kind of process is quite simplified by this CRISPR, and then this is going to be talked about in the next presentation by Mitra Cohen. And then today, I'm going to focus on this third model, quite unique model. Now we can generate using this CRISPR. Those are called somatic cancer modeling. Before I go into those somatic model, it's just to kind of back again. So what cancer modeling in the mice? As I said, we cannot induce the catastrophic change. What we change is introducing a mutation. More specifically, we overexpress oncogene or eliminate the tumor suppressor. There are several ways of doing it. Classic one is kind of treating carcinogen that can be used in the skin or those hepatic tumor. And then once we start to knowing that after molecular biology technique is developed, those transgenic approach is also taken. Several oncogenes identified and putting promoter. If you use a tissue-specific promoter, you can induce the tissue-specific tumor. If you use the ubiquitous promoter, the tissue or the cell type susceptible to those oncogenes start getting tumor. And then also after the discovery of the CRILOX system, you can introduce the oncogene more tissue cell type specific manner, or also the temporary control manner using this LOX flank stop codon with oncogene. Upon the CRI excision, this stop codon is eliminated, this specific oncogene is started to express and then initiate the tumorigenesis. Conditional knockout is also often used. The tumor suppressor gene is kind of flanked by the LOX-P site, and then using specific promoter or the CRI, conditional CRI line, you can eliminate those tumor suppressor. Bill also mentioned about this tetracycline system. This is quite powerful, particularly if you want to study those hormone-regulated organs like breast and then ovary. Because of the CRI-ELT, those conditional system, that is a powerful conditional system, but to induce the CRI activity, you have to use a tamoxifen that interfere with those estrogen activity. It means that those estrous cycles are impacted. So that may affect your result. So in that case, TET system gonna be the way to go. Only downside of this system is you need to have 2 alleles compared to the 1 allele to modulate this activity. That kind of takes another cross to generate the space background. And then those are the germline mice. And then CRISPR editing is adding a kind of option further to this kind of cancer modeling. This is not completely replaced, or rather than that, it's really expanding the tool set. You can deliver the Cas9 on the guide RNA using lentivirus, the tissue you are interested, maybe combine with this germline allele, and then can initiate tumorigenesis. So now we are using this approach, and then that's something I'd like to introduce in the last half of my presentation. But the point is, we are modifying those oncogene on tumor suppressor by overexpression or the eliminating gene to kind of accommodate or to facilitate those final catastrophic changes happen in this tissue. Then a few differences between the traditional germline GMM to the somatic GMM. For example, traditional system, you need to have those specific line in your hand, clear and the overexpressing system, and then tumor suppressor removal. Maybe clear can target, let's say, this luminal cell or those basal cell that you can select if those clears are available. But upon the clear activation, what's gonna happen? All cell expressing clear gonna get this oncogene and then tumor suppressor. Again, this is not sufficient, and then some of them initiate catastrophic change and eventually form the tumor. I'm sorry. This can be starting from a single clone, but it can be the multiple clone. That is difficult to figure out, at least in the traditional system. Instead, in the somatic GMM, you don't need to have a specific line or the specific allele like this. What you need to have is those guide RNA and Cas9 and entire delivery method. So you can deliver these guide RNA Cas9 using virus or the transfection. And then you can deliver those one is directly to the individual cell, and then introduce the mutation. Downside of this system is quite often this cell you want to target should be faced to the lumen or the surface. It will be quite difficult to deliver this system, Cas9 system is surrounded by the other cell or embedded under the other cell. Other difference is compared to the germline system or the systematic system, number of the cell you impact is much less. It's those cells originally mutated is usually surrounded by the normal cell. And also because of the inactivation happened locally, and then this type in left side is inactivation or the activation happened more systemically. Sometimes depend on the clear specificity, you target the uterus, but the tumor emerged from some other tissue because of those susceptibility and clear expression. But this one is locally, always induced locally. Then key component of this CRISPR system, CRISPR-induced Cas9, is you need to have a method to deliver the clear system to the target organ or cell. Several organs has been targeted using various methods. Liver is the easiest one to be targeted using just hydrodynamic tail vein injection. Plasmid inject from the tail vein can be directly delivered into the hepatic cell. Lung, there is a way to deliver intratracheal instillation using a lentivirus. Brain, you have to make a hole in the brain skull, but that is also commonly used technique, intracranial injection using adenovirus. Muscle is you can do the intramuscular injection or electroporation. Pancreas has been used with a few different methods, injection into the pancreatic pancreas using lentivirus, or the retrograde pancreatic ductal injection using lenti, adeno, or electroporation. Breast and intestine, they have a relatively kind of big duct and lumen. They injected those CRISPR component here, delivered those CRISPR using lentivirus or the adenovirus. And Also, not only to the tissue, but also you need to deliver those CRISPR system inside of the cell, target cell. So DNA transfection is a one way. So hydrodynamic tail vein injection is directly delivered to the hepatic cell. Electroporation is another way. And also you can use a polyethylene PEI. Advantage of the transfection is the delivery of the multiple plasmid into the individual cell. Compared to the other virus system, you can deliver more different kind of the plasmid into the single cell. Viral system is quite convenient, has been used a lot, both adenovirus and lentivirus. Difference between adeno and lenti is adenovirus is no genome integration generally. Lentivirus has a genome integration. This has an advantage and a limitation. Those in genome integration may randomly induce a mutation, that is limitation. But if you want to perform those screening, that kind of integration is help to identify the what guide is inducing that particular phenotype. That can be identified those integrated CRISPR or the guide RNA. Both system has some limitation because of the you need to package those virus genome to create virus. So the size of the cargo is limited, low viral cargo capacity. If you wanna do the multiple kind of gene overexpression and then the removal, that is not working well. So to overcome this issue, people already generated Cas9 knock-in mice that is clear control or the uniform, ubiquitously expressed. If you use these mice, you don't need to deliver the Cas9 gene. That is not necessarily small for the packaging. That case is what you need to deliver is just a guide RNA that create flexibility in your system. Third one is a kind of new field, but it has a very strong potential. It's not using those, but now bioconjugate or nanoparticle-based lipid gold, peg, or the other form. That's also including a nano needle or so on. Advantage of this is non-immunogenicity, ability to carry the large cargo load, and the low biosafety demand, so it can be easily used in animal and then easily scalable. For example, one option I always thinking is if you want to target the melanocyte to initiate the melanoma, that is not necessarily easy to target by virus or the others, but those new technology using a nano needle and so on may have a potential to do that. And then I'd like to kind of explain the difference between the germline cancer modeling and also the somatic cancer modeling. Germline modeling, usually we use those conditional allele. For advantage of the conditional allele is always you're gonna make same mutation or the same deletion. Only thing you consider or the Q concern may be homo or heterodeletion based on those clear activity, but once excision happen, that can be generated the same kind of mutation. However, CRISPR-Cas, even you target the same exon, let's say in this case, exon 2, this induces a DNA double-strand break. Each cell has a 2 allele of this exon 2, of course, and then both double-strand break is independently fixed by the non-homologous end-joining DNA repair. Sometimes it's correctly collect. In that case, it's back to the wild type, and then, but in this case, it's retargeted, and then eventually form those indel mutation. Because this is the exon, and then 1/3 can be inflamed, 2/3 can be flame cyst. 1/3 of the inflamed can be either functional or the dominant negative, depend on the where you targeted. Flame cyst is usually the initiated precautious stop codon. The point is those conditional allele always create those consistent mutation, but in those CRISPR-induced mutation is quite valid in the individual allele. maybe mutation 1 is 1 base pair deletion, functional no. 3 base pair deletion mean that 1 amino acid deletion can be functional. 7 base pair insertion, functional, null. 147 is still de-inflamed, but it can be work as a dominant negative hypomorph, and so on. That is unpredictable. That is a difference. And then also this is the kind of our real experiment, just to give you the flavor. We targeted gene called LKB1 using this Cre-conditional array. And also to mark this electroporated cell, we use the Cre reporter. This one has a stop codon and the RFP. After Cre excision, stop codon is removed and then RFP start to express. I'm sorry. And then other 3 tumor suppressor, BRCA1, P10, P53 are targeted by Crispr. And then each cell has 2 alleles, and then each individually or independently targeted. In that means, maybe this one allele can have a certain mutation, but the other allele has another type of mutation, and so on. In each 3 tumor suppressor has a different allele in each cell. What it means is soon after those electroporation or delivery of this Cre and CRISPR, what you're gonna have is a library of a different type of the genotype cells. For example, cell A have those LKB1 deletion or this deletion plus mutation 1 mutation A, mutation 1 wild type, and so on. All cell is theoretically have a different type of mutation. Then, if the mutation is sufficient to initiate the proliferation or some survival advantage, it's gonna form the tumor. Some of the clone progress to tumor formation, some other are not. And then eventually some clone can progress further to the metastasis. This is a kind of difference you can see in the CRISPR model and the germline model. So germline model is constrained by the availability and the specificity of those Cre line, and particularly because of the cancer modeling, you want to have an inducible system. Those tamoxifen induced system and doxycycline system are all nowadays it's available, but not necessarily all cell types. So if you don't find the kind of light Cre line, you need to make yourself, or you need to wait to see if any cell, a good Cre line is available. And also those conditional alleles need to be available. What it means, sorry, keep switching. It's to generate the genetic background you want to test is quite time consuming because you rely on the mouse crossing. On the other hand, a strong advantage is once you establish that your kind of desired genetic background, this is really well established and controllable. Result is quite robust. That is a huge advantage. It takes time to generate it, but once you have it, that you are really kind of reproduce what you want to see. However, only a kind of limitation or the challenge is because of it, to make a certain genetic background to cross the mouse line, it really takes time. So not easy to test various mutation combination. On the other hand, somatic system is, as I keep saying, no need to have a mouse cross or Cre lines or even conditional allele. But you need to develop a delivery of those Cas9 system into the targeted cell population. That you need to be quite creative, but it's depend on that cell type, it's feasible. Point advantage is fast and scalable, and then combined with those germline mouse available, you can make a complex application. And as I said, it's because of the not uniformly induced those oncogenic change, the mutation. The way it's emerged is kind of clonal. It's much similar to the more human cancer. Challenge is editing efficiency is not necessarily high, so you need to come up with a good way of modifying the genome. But huge advantage of this system allow to test various mutation combination. And the downside, particularly if you want to try to use as a preclinical model, this can be the one big challenge. Each mouse is relatively unique. As I told you, that the mutation pattern is we cannot control. They can form the tumor, but the way it's formed the tumor is not necessarily identical. One possibility to overcome this is that yes, once you generate the tumor from the single mouse and then take out this tumor cell and then transplant, kind of similar to the human cancer patient, human cancer model. Okay, now I compared those germline and the somatic CRISPR model, and then we used those somatic CRISPR cancer model to study the ovarian cancer. That's something I'd like to introduce. Before I go to the model, what is ovarian cancer? Is ovarian cancer the fifth most common cause of the cancer-related deaths among women? Oops. And then estimated 3,000 Canadian women diagnosed, and then 1,900 will die from this disease. It's not common cancer, but it's once becoming this cancer, it's highly likely to be died by this ovarian cancer. It's relatively the cancer for the old age women, usually after menopause. And then, but the challenge here is there's no good way to do the early screening and then no specific symptom. Most women diagnosed is quite late stage, stage 3 or 4. Therefore, the 5-year survival rate is quite low, around 27 or 1/3. This is just a simple diagram that, yeah, ovarian cancer is not frequent, but once it becomes the ovarian cancer, most women die because of this ovarian cancer. Compared to the breast and uterus, frequency is higher, but still many of the women can survive and then cure this cancer. And then one type of the ovarian cancer we are particularly interested in is high-grade serous ovarian cancer. This is the 70% ovarian cancer, and then it's quite aggressive cancer. And then this ovarian cancer is kind of unique, not known the recurrent mutation. Mutation is, we believe, the key for the tumorigenesis, but in this type cancer, common mutation is just one. P53 can be seen in about 90% of the patient, but the next one is lower than 5%. So no recurrent common mutation. So what driving this cancer is copy number alteration. This is a comparison. Some tumor are driven by those mutations, some other tumor are driven by those copy number alteration. Ovarian cancer is particularly driven by those copy number alteration. What is challenging in those copy number alteration driven tumor is because of copy number, so chromosome area gonna be amplified or deleted. This case is just I draw the duplication. So what's happening that the 100 or the 200, even more gene on this locus is amplified or deleted altogether. So it is difficult to pin down the important gene or the pathway driving cancer and the metastasis. And what we know by now is roughly half of the ovarian cancer high-grade serous is the deficiency of the homology-directed DNA repair. And then, but some subclass is HLA proficient, but is still forming these high-grade serous. We have no idea what's causing them. And then deficiency genome integrity is really kind of important as a background, but how this deficiency lead to the proliferation and the metastasis is really not known. Gene pathway involved in high-grade serous are not fully understood. Study from the BRCA patient, those patient has a mutation carrier has a higher risk to develop breast and ovarian cancer. And those women has those the BRCA1/2 mutation is kind advised to go the prophylactic bilateral risk reducing salpingo actually, or for rectomy. It means the removal of the fallopian tube and the ovary. From studying these fallopian tube from the high-risk patient, now we call as ovarian cancer, but this cancer is not originated from the ovary itself. It's originated from the organ next to the ovary. It's called fallopian tube epithelium. So this is just a simple diagram of the female reproductive tract. Ovary is here, and then those uterus. Fallopian tube is this tube. Its normal function of the fallopian tube is a place for the fertilization. Epithelial lining on this tube is somehow transformed through this kind of changes in forming a precancerous lesion, and then eventually spread to the ovary and also to the peritoneal cavity. So because of the original people thought the ovary is the origin, and then suddenly switched to the fallopian tube is the cell of origin. When we start working on this ovarian cancer project, there is no good clear line available. So in that case, two choices. If you want to make those traditional germline mouse model, you need to generate a clear line. To make a clear line, you need to know what gene is specifically expressed in this fallopian tube. That it takes lots of time. So instead of making those germline system, we decided to go to this somatic system. So what we did is we delivered CRE and CRISPR plasmid inside of this oviduct lumen by micro-injection, And then once we filled those DNA solution into the lumen, and then using those tweezer type electrode, and then grab the tissue and then zap it. This is a normal electroporation, so all the cell lining on this lumen get, not all, but some of the cell on the lumen get electroporated. It's forming a somatic germ, GEMM. So we targeted 3 or 4 tumor suppressor genes based on the literature or based on our interest. I'm not going to go into too much detail of each tumor suppressor, but what we targeted is P53, P10, BRCA1, and LKB1. And then also we used those CRE reporter to track the electroplated cell. Upon the CRE delivery, those cell, oviduct epithelial cell, is those stop codon is excised and then initiate the RFP expression. So we can track this cell as an RFP positive cell. Here is a proof of principle. So we inject and then apply the pulse, and then so that initiate those CRE excision, and then initiate the RFP expression. As we expected, luminal epithelial cell become RFP positive, and this is a high magnification. Not all the cell, but those cells, some of the cells are becoming RFP positive. That can stay in the several months. And also, this is sufficient to introduce a mutation? Yes, it is. So we collected those RFP positive cells using FACS sorting, and then checked the mutation efficiency. P53 allele, BRCA allele, P10 allele. Those RFP positive cell has all, at the P53 locus, it's all mutated. P10 allele all mutated. BRCA1 is a little bit lower, but 80% is mutated. Same thing is LKB1 allele is controlled by those flux on the CRE. So LKB allele is deleted into this RFP positive cell, and then flux allele is retained in the RFP negative cell. That's expected. So we can deliver the CRE and then CRISPR, and then initiate the introduce the mutation to the target gene. Long story short, this is sufficient to create high-grade serous tumor in mice. They formed a kind of high penetrant ascites similar to the patient, and then create the kind of characteristic peritoneal metastasis similar to the human patient. Here is a kind of number because we can play those gene mutation number. When we mutate 3 genes, it takes relatively long latency, roughly 1 year, and then penetrance to the tumor genesis about 20 to 30%. However, when we combine with the 4 tumor suppressor gene, and then it can form the high penetrance and the short latency. And then back
Dr.
to the
Yamanaka, you have about 5 minutes left.
Yep. And then so to confirm those mutation pattern, so we checked those targeted sequence. This is the one target sequence of the P10. And then when we analyze from the single mouse, we find the 6 types of mutation. We can place back to the original tumor. This experiment, we targeted the left and the right ovary, those fallopian tube independently. But we found the mutation 1 to 5 can be seen the only left side, and the mutation 6 is only right side. And then what is interesting here is that in the metastasis, you can find only mutation 6 and 1. So mutation 2 and 5 is not selected or not progressed to the metastasis. Because of the we can track the RFP, we can see the micro met in the body and then the mouse predatory cavity. That is an advantage. I'm sorry. So this is the original tumor is small and 5 millimeter, 1 centimeter, but already metastasized to the peritoneal cavity. Because of the RFP, we can see in those tiny tumor on the peritoneum can be trackable. And I just want to kind of skip here. So CRISPR-mediated somatic cancer modeling it is easy to change the mutation combination. This is a huge advantage. NHEJ mutagenesis can easily use for the gene deletion. Point mutation can be introduced HDR or the base editor. That HDR is efficiency is going to be the issue. Base editor is one kind of interesting direction people should try. And then overexpression system is one kind of interesting direction. We are kind of developing a new technique using a CRISPR-mediated insertion or the transposon. And then, of course, we can combine with the already pre-existing CRILOX system mouse line. And then some people try to kind of initiate a locus amplification or the deletion. So chromosomal modification is not high frequent, but can be initiated. Chromosomal translocation or the huge deletion can be induced by this CRISPR system. Tracking clone are possible retrospectively by sequencing of fluorescence reporter. And then another important point, the tumor is always developed locally. So here is a kind of acknowledgement. The most of the data generated by KP10, and now she's in Toronto, but yeah, all the others supported to generate those ovarian cancer models. And then these are collaborators. Yeah, thank you.
Thanks so much, Yojiro. This was really the whole perspective of what can be done, and very informative and very loaded with information. So I'm sure, do we have any questions in the chat for you at this point? It doesn't seem like it. One question that I've been toying with many times, what is the maximal insertion that you can remove by placing LOX sites further away? I mean, on one of your slides, it said 147 base pair. Can you go long, even?
That one is the non-homologous end joining. So that's something quite important. People think that those non-homologous end joining create a small indel mutation. Small indel mean that maybe a few base pair deletion or the insertion is the most common. That is true, but NHEJ also initiate long deletion. That's always you need to keep in your mind, because of the most of the time when people try to detect NHEJ, we use a small PCR reaction, maybe 100 to 200 base pair PCR. But occasionally, those NHEJ can go beyond this PCR For example, if deletion is bigger than your primary primer annealing site, you won't detect it. That is always you need to keep in your mind. That is one point. For example, that is why occasionally big deletion happen, but sometimes you may miss it. And then how long is we can control the deletion? That is going to be the one issue, or the bigger the deletion, probably lower the frequency. And then I haven't tested myself. Maybe Mitra may comment a little bit, but it's reasonably few kb seems to be efficiently deleted using a 2 guide RNA.
Yeah, so I'll talk about this, but we've done up to 5 kb, so you can do multiple kb with this.
Okay. So we are a little late on our schedule. I suggest...
Hey, Nicole, we have a question on the chat that maybe we can just...
Okay. Do you want to ask the question? Okay. So it says, thank you for the talk. It was very insightful. Just curious if you mentioned whether you compared the tumors generated in your model to clinical ovarian cancer samples from TCGA or local samples you may have access to.
Yeah, these ones, I believe they're talking about RNA expression or those genomic pattern that's we are currently working on. And then, yeah, that's we are working on. Pathologically, it's really similar to the high-grade serous, but RNA and those genome changes, we are now checking.
Peter, you have a question, I think, eh?
Yeah, Yojiro, that was very, very nice. So you have these 2 different patterns of metastases, right? These really flat-looking metastases versus these ones that seem to grow out of the tissue a little bit. Were you seeing any differences in the spectrum of mutations in either of those 2 types of lesions? Do you think that that's driven by a mutational combination or something else?
That's something really we want to answer. And then, but generally speaking, it's not just mutually exclusive. It still can be, there's a spectrum, but it's not exclusive. And it can be seen both in human and then also our mouse model. What causing it right now, I don't know. Yeah.
Okay. Very nice.
Okay, so considering the human body was not designed to sit in front of computer screens for 6-7 hours at a time, we will take a 10-minute bio break